Welcome to the Neuroprogressive and Dementia Network strategy launch. So I'll just give a little bit of preamble. As you can hear, I'm suffering a bit from a nasty cold, so I'm just going to do the introduction, and then Peter is going to invite speakers to come to the podium here. It's a very mixed conference. There's quite a few people doing their presentations online, which, because of the Omicron, I had to write it down because I can't remember, new variant virus, we had to change the conference at the last minute and give the opportunity for people to not be here in person. So we're trying to encourage people to wear masks when you're sitting at the tables, and just be very mindful of all the hygiene, and try and space yourself out whilst you're sitting here. So it's great to see, it's great to see a good group of people here in person, and I think there's 107 people have signed up for online. We've got some really great speakers lined up for you. We have a poster prize of a £50 voucher, which Peter Connolly and Vivek Patton are going to judge for us, and that will be announced later on. So anyway, enjoy the day. Thank you very much for coming and for being online. So Peter is going to introduce the speakers. Thank you.
So, yes, good morning, everybody. So my pleasure initially is to introduce Dario Alessi. Now, Dario is the director of the MRC protein phosphorylation and ubiquination unit, and the Dundee signal transduction therapy unit, which involves a lot of collaboration between his extensive research and the pharmaceutical industry. His a lot of echo. His interests are very extensive and cover a number of disease areas, but particularly relevant to us is work on understanding the LARC2 variant of people, a genetic variant of people with Parkinson's disease. So I will pass over without further ado. Dario is doing his presentation online. I'll pass over without further ado to him.
So thanks so much, Peter and Neil and others, for inviting me to present this lecture. So today I'm going to tell you about, not just my own research, but a more general talk about the effort that's going on in the University of Dundee to study Parkinson's disease. And at the moment we have a group of 8 researchers who are shown here. 3 are consultant neurologists, Miratul, Esther and Tom Gilbertson. And then we have 5 basic researchers, myself, Satpal Gopal, Ian Ganley and Andy Howden. And 7 of us are based in the same building. 6 of us are based in the same MRC, unit. So we work in the same laboratory space. So we essentially talk to each other nearly every day, and our students and postdocs, work between groups. And, we're very inspired between having the basic research and the clinical research going on in the labs adjacent to each other. And it's, it's wonderful for us to, to be able to talk to clinicians and also the patients that the clinicians often see. And, just to say, we engage tremendously with patient groups and their families, and we have this, very active Dundee research interest group, especially where, we talk with them, most weeks or months. And, that's very inspiring for all of us to, to continue this research in a very difficult, research area. And we're also very grateful to the University of Dundee because they've recognized Parkinson's disease as an important area to support. And the university's actually been organizing on our behalf, quite significant, Parkinson's research campaigns that, have raised, very substantial funds of money, and that are greatly appreciated to enable us to, to do more important research, and also, bring young scientists into our labs to get, internships and training for, their research. So I think you'll all know in this audience what Parkinson's disease, how it affects, people, and and it's. I'm told it's approaching 10 million people in the world now are living with this disease, and it's the fastest growing neurodegenerative disorder. And yes,
your slides are not moving forward, I believe. You're not in presentation mode.
They are for me. You don't see them moving forward.
Not on my screen.
No, nor on mine, Dario.
Oh, that's very bad. They're moving forward. Maybe something's gone wrong. Maybe.
No, that's it.
Oh, you can see them moving forward here, can you?
Yeah.
Okay, maybe I'll stick to this format. I apologize. Those were the slides of our patient groups I showed. These were the slides of our, investigators. And this is the slide on Parkinson's disease as I'm talking about. Okay, thanks for letting me know about that. And and I think also what really motivates us is that all of these treatments that help the some of the movement disorders. There's nothing that arrests or slows even the progression of the disease. And this is really the motivation of our work, is to really understand the disease sufficiently that we can work with clinicians and pharmaceutical companies to help develop new therapies that will slow or hopefully halt the progression of the disease. So our work largely focuses on the genetic causes of Parkinson's disease, because in these cases, and it only represents about 5% of people with the condition, at least with this form of the disease, it's certain what is the gene that is triggering the disease. And then this enables us to, understand the cause of the disease. And our philosophy is, if we understand how these genes are controlled and how they work, and what goes wrong in people who have mutations in these genes, and how this disrupts biology to cause Parkinson's disease, we can then play the engineer and come up with new ideas to counteract these disease-causing changes, and hopefully help develop new ways to treat and also diagnose the disease in the future. So in Dundee, there's at least 5 genes that we specialized in. There's about 20 known genes that are involved in Parkinson's disease, and we focus on 5 of them that are highlighted in this slide. And I'm going to say a little bit about LRRK2 and PINK1 and Parkin, and I won't have time to discuss the other genes, but obviously please do get in touch with me if you're interested in these ones. So LRRK2 has been the focus of a lot of our research. We started working on this enzyme in 2004 when it was first discovered to be involved in Parkinson's disease, and this was our first entry in the University of Dundee into Parkinson's research. And I guess all the work we've done with LRRK2 has branched out into a lot of the other things I'm going to talk to you about today. So the reason we're sort of interested in LRRK2, it's one of the most common genetic causes of Parkinson's disease, and I'm told around 1 to 2% of all Parkinson's patients in the world have an LRRK2 mutation. Obviously, in some regions, Ashkenazi Jewish and North African Berber populations, that maybe even the majority of people with Parkinson's diseases in these regions will have LRRK2 mutations. But it follows a disease course that's very similar to the common sporadic form of the disease. So we're suggesting that this biology might be impacted also in the common form of the disease. And so in terms of the biological work that we've been doing, the key aim of our work, when we started this project, was to identify what the role of LRRK2 was in biology. So LRRK2 encodes an enzyme that's called a protein kinase. These are enzymes that regulate all biology. They reside in, they control pathways that control important biological processes. And the way these enzymes work, kinases, they attach phosphates to specific substrate molecules. So the key was to identify what the substrate was for the LRRK2 kinase. And to do this turned out to be incredibly difficult. We started in 2004, and by 2010, we had got nowhere in this pathway. And everybody was saying to us we should maybe find a new project to work on. But our philosophy was to actually, rather than giving up, was to put more effort into the project. So we decided this project was so complicated, we need to set up a huge collaboration of teams of different expertise that we needed to solve this problem. So we worked with 4 other research groups who had distinct and complementary expertise that we didn't have in our unit. And we also worked with 2 pharmaceutical companies who gave us drug-like molecules and also some very sophisticated animal models, mouse models, that were needed for the research. And this was sort of very heavily supported by the Michael J. Fox Foundation, and the key scientists in this foundation were also involved in the project. So this took another 6 years of painstaking research. But in 2016, we identified what the key substrate was. This is another enzyme called the GTPase. And this enzyme also is a key regulator of biology. It sort of regulates vesicular biology, membrane homeostasis. And LRRK2 phosphorylates this enzyme in a key region that is in the middle of its sort of the business end of this molecule that works to recognize other proteins when it's bound in the GTP binding state. And this finding was very important because it gave us the first way to study the function of LRRK2 in cells. And we quickly developed an assay to look at Rab protein phosphorylation in cells. And what was super exciting was that we found that in control people or cells who don't have from cells derived from people or animals without LRRK2 mutations, the levels of Rab protein phosphorylation are moderately low. But when you have the Parkinson's causing mutation, you get increased phosphorylation of the Rab protein, indicating that the mutations stimulating the activity of LRRK2. And then when you treat the cells, in this case with a LRRK2 inhibitor developed by GSK, you can see you lose completely the phosphorylation of LRRK2. There's about 20, we know of 23 mutations now that cause Parkinson's disease in LRRK2, and they're all in different regions of the protein. I don't have time to go into this in this talk, but it turns out that all of the Parkinson's causing mutations are likely to stimulate the activity of LRRK2, which means that inhibitors or drugs that target LRRK2 might have benefit in counteracting the mutations. And this is one of the most exciting therapeutic avenues that is being explored by drug companies. And we now know it's a bit more complicated. There's a whole dozen or so Rab proteins that are key substrates of LRRK2, and these are phosphorylated, and the phosphorylation of the Rabs are probably involved in mediating diverse and complicated biology. And around this stage of the project, we're talking around 2016, 17 now, Esther Samler became very interested in LRRK2. And, she wondered whether she could study LRRK2 in human patients, because most of our work was done in cell lines or mouse models. And, Esther set up a very sophisticated assay where she can now measure this Rab protein phosphorylation in white blood cells derived from patients. And these are either neutrophils or monocytes. And Esther's shown very nicely that people with mutations in LRRK2, you see an increased phosphorylation of the Rab proteins, in their blood cells, and you see this also with other mutations, for example, in the BPS35 pathway that also activates LRRK2. And I just want to show you one recent data that Esther showed me on Wednesday this week. So he's doing a screen of 100 people who have inherited Parkinson's disease, but they don't have a mutation in LRRK2. This is known from the genetic sequencing. And he's screening these patients to see if any of those have elevation of Rab protein phosphorylation. And in this screen, I think he's identified about 6 to 10 people who have very high levels of Rab protein phosphorylation, but these people don't have mutations in LRRK2. So this is one of the hits that is seen in this screen. You see a 16-fold enhancement of Rab protein phosphorylation. So this is really exciting because it suggests that there's potentially other genes that can be mutated that could cause Parkinson's disease, that could also activate the LRRK2 pathway. And Esther hopes to, identify, these genes with her collaborators. And, so because LRRK2 activates, because the mutations that cause Parkinson's disease activate LRRK2, our work and others have stimulated a lot of pharmaceutical companies to embark on developing LRRK2 inhibitors. Many companies are now working in this area, and at least 2 are in clinical trials. There's probably more that haven't yet announced that they're in clinical trials. But the company that's most advanced is a company called Denali Therapeutics, and then they're just about to initiate, in a collaboration with Biogen, a clinical trial that's going to cost over $1 billion to undertake. This is a worldwide trial. It's going to be sort of the late phase trial, phase 2, phase 3, where they'll treat both people with sporadic disease and another group with LRRK2-driven disease to test their inhibitors. And Esther hopes that we're going to get hopefully we'll be able to bring the clinical trials for these compounds to Scotland, because this is very important for us, because it would enable our Scottish patients to participate in these trials. And Esther's working hard to try and achieve this. So I'll go on to our other work. We obviously now understand a lot more about the LRRK2 pathway and how it works. And but for the interest of time, I'll just talk about some of my other colleagues' research. Mirator Muckett, and Ian Ganley are working on these 3 genes, such as PINK1 and Parkin. But Ian's just discovered that LRRK2 also plays a role in a process known as mitophagy. So it turns out that mitochondria, these are the key batteries of the cell that make a lot of energy that the cells and the brain cells need to survive. And as a natural process, these batteries become degraded, and they have to be removed from the cell by a process called mitophagy to enable new mitochondria to be formed and replace them. And it turns out that many of the genes that cause Parkinson's disease are regulating this process of mitophagy, especially the PINK and Parkin pathway. And what Mirator has discovered in beautiful work is that when you have damaged mitochondria that become old and need to be replaced, this leads to a huge activation of this enzyme called PINK1. And then this actually becomes stabilized and binds to the damaged mitochondria. And at these locations, it phosphorylates proteins like a Parkin, that's another Parkinson's disease gene, and activates it, and also phosphorylates ubiquitin, another protein called RAB8. And through a very complicated pathway that's not fully understood, this induces the damaged mitochondria to be degraded through this process known as mitophagy. And my colleague Ian Ganley has discovered this fantastic new assay that's called MitoQC to study mitophagy. So in this assay, it's a very interesting assay. He attaches to mitochondria a red and a green protein. So that means the mitochondria, red and green together, if you mix the colors, you get yellow. So the mitochondria are all yellow. But it turns out when the mitochondria is being degraded, the acidity of the vesicle that it's in it goes down, and this quenches the green color, but not the red color. So the mitochondria that are being degraded turn red. And so this assay has revolutionized our ability to study mitophagy in cellular organisms, including in dopaminergic neurons. Now Ian is able to study this process. And this has led to tremendous advances in our understanding of how Parkinson's disease control mitophagy. And in a recent paper, Ian's actually very unexpectedly discovered that LRRK2, when it's activated, sort of inhibits mitophagy through a pathway that we don't know the mechanism yet. But this is a very interesting result because it suggests that LRRK2 inhibitors will relieve this inhibition and therefore stimulate basal mitophagy. And and then this is also that LRRK2 inhibitors might be beneficial for patients, because that would be a very good thing to do to stimulate mitophagy. My colleagues Gopal Sapkota and Sapal Verde are working on chemical probes that can change the activity of key enzymes in the pathways involved in Parkinson's disease. And Gopal's working on a technology that's best described as developing sort of chemical missiles that can attack specific proteins that are toxic in cells and induce them to be degraded so they no longer become toxic. And his work's focusing on alpha-synuclein, which is another very important protein in Parkinson's disease. And many people with Parkinson's disease have abnormal levels of aggregated alpha-synuclein in the brain. And this work's complemented by Sapal, who's developing probes against E3 ligases that might help with targeting of these toxic proteins. And I'm just coming nearly to the end of my talk, and Andy Howden is our youngest colleague in this collaboration, and he's a specialist in a technology called mass spectrometry, and he's basically trying to use this technology to identify specific biomarkers. So at the moment, patients are just told they have Parkinson's disease, but what Andy hopes to do is to be able to use these new biomarker technologies to try and group patients into different groups to understand how the disease is driven, and whether you can work out which patients the disease might be driven by pink or parkin, which one's alpha-synuclein, which one's by LRRK2, and then that might help group the patients into groups that might determine which might be better treated by different therapies. And finally, this is the hardest one for me to explain as a biochemist, but I'll try and do my best to tell you about the work that Tom Gilbertson is doing, which is incredibly spectacular. And he set up in Dundee this MR-guided focused ultrasound, neurosurgery methodology. And this is only the second treatment center in the UK that offers this technology. And essentially, people with at the moment it's being used for essential tremor, but I think he's getting or received approval to also use this for trials in people with Parkinson's disease. And the aim here is to use focused ultrasound in an MRI machine to ablate tiny millimeter spheres in the brain in specific regions or circuits that cause involuntary shaking and and movements. And Tom kindly sent me a video of his first patient that he's treated, and I think he's treated half a dozen or more people at the moment with this condition, and the patient's given consent for the video to be shown. So this is before the procedure, and you can notice this gentleman, he has a very bad shake in his left hand, but he also has a minor shake. You don't really notice it in his right hand. Okay. And the way this method works, I mean, apologies, I'm a biochemist here. It's only safe at the moment to target one area of the brain. So they only do one of the brain, and then they're going to wait a year before doing the other side. So this patient's only been treated on the side that controls the tremor in his left hand. And you can see after the procedure, the patient, you see he's lost his tremor in his treated side, and it actually his other side looks much worse than it did in the previous video. And obviously in a year's time the patient will be able to be treated in his other side. And these treatments are revolutionary for these patients. Apparently, one person I heard about hadn't been able to pick up a glass of coffee for 40 years, and immediately after his treatment, he was able to pick up his cup of coffee. So that's really quite remarkable, and hopefully that will benefit people with Parkinson's disease in the near future. And obviously I thank in this slide a lot of people who've worked with my team, especially over the last few years, and I highlight a lot of our very generous funding sources that come from the MRC, the Fox Foundation, this new foundation called Aligning Sciences Across Parkinson's, and also obviously Parkinson's UK. And happy to take any questions.
Are there any questions from the floor for Daniel? Or anything that we've had on the chat? No? No? Well, you're getting off lightly, Daniel, so you'll be delighted. So once again, I think Dario's talk shows the real value of persistence. When you feel that you have a breakthrough around the corner, sometimes it's not just around the corner, but I think we'll all be grateful in a few years' time, certainly, in the hope that the LAR2 inhibitor trial does actually do what the science would suggest that it does, and that we have an effective treatment to slow down the course of Parkinson's disease in due course. So once again, can you just join me in thanking Dario for his talk? Okay, and then next up we have our colleague Suvankar Pal. Suvankar is an academic neurologist who works at the Anne Rowling Clinic in Edinburgh, in addition to working in the NHS Forth Valley. It's a very interesting mixture of clinical and academic work. And he has also a number of strings to his bow, including the CJD Surveillance Centre, but importantly he has been one of the co-leaders on the MND SMART study, which is looking at some repurposed drugs that have been used in other conditions, which might be effective in either slowing down the deterioration of motor neuron disease or perhaps even having an effect on survival. So I shall pass over to Suvankar for his talk.
So, really by way of background, motor neuron disease is a rapidly progressive but unfortunately fatal neurological condition. From when people first develop symptoms to when they pass away, it's unfortunately very brief. So we're talking about 2 to 3 years from symptom onset to death. And during this time, they experience a host of very challenging symptoms, including weakness in the arms and legs, difficulties with speech and swallowing, and ultimately people die from failure from breathing. We understand increasingly that people with motor neuron disease have cognitive behaviour change, and that this affects up to 50% of people living with the condition, and 15% of people have frank dementia, frontotemporal dementia. It's a difficult diagnosis to make, and people often have symptoms for up to a year before the diagnosis is clinched. And thereafter, about 1 in 3 people die within the first year. We understand that about 1 in 10 people have a genetic cause for their condition. And whilst it's thought to be a rare condition with 5,000 people affected in the UK, when you think about the lifetime risk, the reality is that about 1 in 300 to 1 in 350 people will develop MNT in their lifetime. So it's not insubstantial. Against that background, there's only been 1 licensed treatment available in the UK for people with MNT, and this has been the truth for the last 25 years. So since 1995 we only have 1 drug that we can offer as clinicians to people with MNT, a drug called Ozol, and this medication only extends life by 2 to 3 months. There have been other medications that have been licensed in countries like Japan and the USA, but not in the UK. And the reality is that in over 125 trials in the last 10 years, that these have failed to identify alternative treatments. So clearly the unmet need is for innovation in how we select drugs, innovation in clinical trial design, and improved participation of people living with MND in trials. The sad reality, again, before we launched NDN Smart, is that the number of people who had participated in trials in MND in the UK was less than 5% of people living with MND, and in Scotland less than 1% of people living with MND. Why is this the case? Why have we had 10 years of trials, over 100 trials, but no specific treatments? We can kind of think about it in three different ways. There are biological factors, limitations in the way that trials have been delivered and designed, and patient-related factors. We heard from the previous speaker that there are lots of advances being made, but the harsh reality is that we still have an incomplete understanding of the human brain in disease, and therefore an incomplete understanding of therapeutic targets. And linked to this are poor preclinical models. We're trying to extrapolate a lot from poorly based animal models and other preclinical models into what is actually happening in clinical practice. There's also been an inconsistent approach to drug selection, and we know that when we have drugs in clinical trials, we need more sensitive and specific biomarkers to assess the efficacy of drugs. Now, one of the limitations of trials over the last decade has been that the number of individuals in those studies has been too small, so they've been underpowered. The inclusion criteria have been too narrow. The outcome measures have been too blunt. There's also been a real problem with having centres with expertise in running trials and infrastructure and expertise in staffing and the confidence to be able to recruit and assess people with neurodegenerative conditions, and particularly MND. And then often people with these vulnerable neuroprogressive disorders have problems attending appointments. We have quite intensive follow-up schedules, and all of these things lead to high rates of attrition. And then if you think about the actual individuals living with these disorders, I mentioned there's often a diagnostic delay, and by the time people are diagnosed, they're often quite far advanced into their symptoms, and then symptoms progress rapidly. So there's a narrow window where they might wish to take part in a clinical trial. People with MND in particular have difficulties with speech and swallowing, and actually being able to swallow a tablet might be difficult. So again, all of these things lead to high rates of attrition. So what we've tried to do to overcome this in our study design is to really develop an interdisciplinary team that extends across drug discovery and disease biology, people who have expertise in trial design and clinical statistics. So in particular, a number of statisticians from across the UK, including the MRC Clinical Trials Unit in London, and Max Palmer, who has led a number of trials in cancer medicine, including the Stampede study, as well as statisticians from Warwick and Edinburgh. Drug discovery, including stem cell biology and other ways of rapid screening of pharmaceutical compounds. And also people with expertise in difficult, can hear me? Neurological trials. So people who have run trials in MND and in multiple sclerosis and in other challenging disorders. Crucially also, what we've done is involve people living with MND in the study design. I've mentioned there are high rates of attrition in historical trials. How can we overcome this, and what do people with MND want in terms of how they can contribute? The reality again is that the majority of people with MND do want to contribute and do want for people to be able to come up with a treatment and a cure earlier, and if not help themselves, then to help others. What they uniformly fed back to us was that don't use drugs in tablet form if possible, reduce the number of appointments or travel time to clinic, and if you can you consider capturing information that's relevant in other ways using medical technology. So listening directly to them, we incorporated a number of design factors in MND Smart, including use of liquid IMP, with courier medicines to people's homes, and even pre-pandemic, we had in the protocol that we would assess people remotely using video calls. We're working in sub-studies on using wearable devices and things like voice acoustic data capture. So the study MND Smart is a systematic, multi-arm adaptive randomized trial, and most people will need no introduction to randomized trials. The novelty of MND Smart for a neuroprogressive disorder is that it is a multi-arm trial. We'll be looking at more than one investigational product at the same time. And it's also adaptive, which means that we'll be looking at all of the accumulating evidence as the trial is progressing. And we have interim analysis points at which treatment decisions will be made about whether a drug continues in the trial or whether we stop because there's a definitive data set that demonstrates the drug is not working. And as Peter said, the initial focus is on repurposed drugs, so these drugs that have been shown to be safe and effective in other progressive disorders, but these are disorders with shared pivotal pathways, and there's a good evidence base for using them in MND. But our aim is to rapidly move to evaluating candidate medicines from discovery research. And the reason we're adopting this approach is that promising new treatments are being identified rapidly because of advances like stem cell biology and the ability to emulate the disorder from individuals with MND and generating their stem cell technology. And so we need to evaluate these definitively. We also understand, and as I mentioned, historical MND trials have been ineffective and have demonstrated that the majority of drugs are ineffective. So we understand that the majority of drugs that we try will also be ineffective, and we need a rapid way of determining this. So rather than testing treatments individually in 2-arm trials that run for 6 months and show negative results and then re-establishing infrastructure and starting again, we're adopting this so-called MAMS approach, multi-arm, multi-stage, which will test drugs efficiently and definitively. And the adaptive approach that I mentioned, which allows us to look at the data as we're accumulating it, allows ineffective drugs to stop early and new treatments to be investigated in a timely way. How are we selecting our drugs? Well, there are a number of different approaches. One approach is to look at clinical and preclinical studies that have been done in other neurodegenerative disorders in a systematic and unbiased way, and this is working with the Camarades team led by Malcolm MacLeod at the University of Edinburgh. And this generates a long list of drugs. So we look at disorders that have shared pivotal pathways, so the dementias, Alzheimer's and frontotemporal dementia, Huntington's, motor neuron disease, and progressive MS, as well as Parkinson's disease. And we looked at clinical and preclinical studies, that includes animal and human stem cell studies. And then we ranked all of the studies in terms of their efficacy, safety, study size and quality. And this generated a long list, and then we had an expert panel that reviewed the list that was made up of clinicians and biologists to think about the feasibility of moving to trial and also not investigating drugs that have been looked at several times before. Now we can finesse this approach now by looking at mechanism of action and pathway analysis studies. So when we have the long list, we can run those drugs in the lab through that, and also the high throughput phenotypic drug screening and cellular models of MND, and that helps nuance our drug selection. And so this is the methodology that we'll be using moving forwards for selecting new drugs in MND Smart. The first 2 drugs that we're trialling are memantine and trazodone, which will be familiar to the audience here. Memantine is a non-competitive NMDA receptor antagonist, which is licensed for use in moderate to severe Alzheimer's disease. It has a neuroprotective evidence base in vitro and in animal models of disease linked to NMDA and glutamate-induced toxicity. There's good evidence from preclinical models of MND, but we don't really have sufficient evidence from trials in MND. They've been underpowered. Crucially, there were no adverse effects, increase in adverse effects in those trials. This was a good candidate drug to start with. The second drug that we're trialing is trazodone, which again, you'll be familiar with as an antidepressant. We're not so interested in its serotonin properties, more in its PERK inhibitor enzyme, so the protein kinase RNA like endoplasmic reticulum kinase, which is pivotal to stress granule formation. So this drug is licensed as an antidepressant. It's used in vulnerable people already. It's safe. There is good evidence from preclinical models that it would be neuroprotective, and it's been trialled in small numbers in dementias and Parkinson's disease, particularly looking at behavioural and non-motor manifestations, and improvements in cognition, mood and sleep have been reported. And so individuals are started on liquid IMP at quarter dose and then titrated up 5, 10, 15 and 20 milligrams of memantine. And those of you that work with people with dementia will know that this is the standard sort of dose. And for trazodone, for quite a low dose, really 50, 100, 150 and 200 milligrams is the dosing escalation. What about outcome measures? How are we going to measure if these drugs are working? And how are we going to get the regulators to approve the drugs if there is a benefit in the trial? The majority of trials historically in MND have looked at functional assessments. So the ALS-FRS is the industry standard, rating scale used in MND, the amyotrophic lateral sclerosis functional rating scale. And this is quite a simple test. I mean, one of the problems we have in neuroprogressive disorders is that the outcome measures are kind of crude pen and paper based questionnaires that we administer at intervals through the study. But functioning is important. The gold standard is survival, and MHRA and EMA are interested in survival. The difficulty with survival is that you require large numbers of individuals and longer duration follow-up, and potentially it misses the benefits of therapy on functioning. So I mentioned Riluzole extends life by 2 to 3 months, but there's no real evidence that there's any quality of life that's improved. So what we've opted for is a primary objective that has a co-primary outcome of ALS-FRS and survival. And we're going to look at ALS-FRS in the initial stages of the trial, stage 1 to 3. And if a drug passes through these stages, conditional on its functioning, we will also progress to stage 4 to evaluate survival. It's important to look at secondary objectives, and the MHRA did push us really to say for a devastating disorder like MND, you should be thinking about things like quality of life and cognitive functioning and mood as well, and particularly given that the first 2 repurposed drugs that we're looking at have got functioning benefits for cognition and depression potentially. They asked us to look at those too, so we are with standard rating scales. So this cartoon summarizes the study design. We have 3 arms to start with, 2 active treatment arms and 1 placebo arm, standard of care. And individuals will be followed up, and then there'll be interim analysis points. So the first interim analysis will occur at stage 1, and that's when 50 participants have completed 6 months or more in the study. And at that point, we'll look at a change in the clinical rating scale, the ALS-FRS. And then if individuals pass that predetermined point that we've set, they'll move on to stage 2 and so on, stage 3, and then survival analysis. If a drug is not performing well, there's a definitive stop at each of these points, and we have the ability to introduce new drugs. How are we going to recruit patients? Well, just a little note about the Scottish MND registers. This was the first international register for people living with MND, set up by Rob Suvankar and Charles Waller back in the late 1980s, and then rebooted by ourselves in the last decade. And we have achieved ongoing comprehensive monitoring of all people living with MND in Scotland. We have a very high rate of ascertainment of 99% of individuals, and we now have comprehensive clinical annotation and follow-up of people from diagnosis to death across all 14 health boards. And through Scottish Government funding, we've doubled the number of MND nurses and have a ratio of 1 nurse to 40 people living with MND, which helps with all of this. And people with MND over the last decade have taken part in a number of observational studies and bioresourcing studies, but crucially what they're interested in is taking part in clinical trials. There are 450 people living with MND in Scotland, and about 80% of those have registered interest in participating in a study. However, we also needed to reach out to partners across the UK, and when we launched the study in January of last year, we had a huge amount of media interest on the BBC 6 o'clock news and the 10 o'clock news on that evening, and over the course of the next few weeks and months, 1 in 4 people living with MND in the UK have registered their interest in participating in the study across the whole of the UK. And so we initially planned to have 15 sites opening across the UK. Obviously, we were hit by the global pandemic, which proved a challenge. We actually opened in February 2020 with our first participant in Edinburgh, and we recruited 7 people before the first lockdown. And at that point, recruitment into all clinical trials was suspended for a few weeks. However, as I mentioned, our protocol allowed us to do remote assessments, and so we learned quite a lot by following people up for the first few weeks of the study, and managed to have some protocol amendments that worked very well when the pandemic allowed us to recruit again. And in line with essential healthcare, we were then allowed to recruit people into the study, understanding that there are no viable treatment options really for this group of individuals. So very quickly after that, over the course of the summer and autumn last year, we opened across the whole of Scotland, and then in other sites across the UK, and we've continued to open this year. Just last week we've opened in Wales as well, in Cardiff, so I think, I lose count, but I think we're at 17 sites now in total across the UK, ranging from Inverness to Exeter. And it's been a great experience working and partnering with these sites. They've all been extremely enthusiastic. They've all have patient populations that have been keen to take part in studies. They haven't historically had the opportunity to take part in clinical trials in MND. There have only been 4 or 5 sites in the UK that have recruited historically. I mentioned that less than 1% of people in Scotland had taken part in the MND trials and less than 5% in the whole of the UK. So it's been something that has been driven by patients, driven by clinicians, and we've been very honoured to work with partners across the country. What that's also meant is that we've been successful in recruiting people despite the pandemic, and we've now recruited, I mean, it's very easily the largest number of people into a single MND study in the UK now at 240. I think we're at 242 from when I submitted the slide deck last week. And this has been, you can see the numbers here. We reached our 100th participant in Edinburgh a couple of weeks ago. Tayside opened up shortly after us. I know the team are here today, and they were really supporting us as the second major site in the UK, and across really major metropolitan areas. But a key approach that we took was that we weren't just going to go for major university hospitals. We've partnered with areas like West Suffolk and Inverness and places like Ipswich. So every individual with MND should have the opportunity to take part in a clinical trial. That was our ethos. It shouldn't be that you have to drive past 2 or 3 different hospitals to get to a site. And actually, colleagues have managed to recruit people from Orkney and the Isle of Wight. So this has been a first for a neuroprogressive disorder. Key to the success of anything like this is support from our funders, and so we've been funded by the Ewan MacDonald Centre, MND Scotland and the Doddy Weir Foundation, and all 3 have been extremely supportive throughout the whole process and continue to be so. So what this has meant is that despite the pressures exerted by the pandemic, our recruitment target is pretty much on track for where we'd like to be at this point. Just to share with you some baseline data from the last independent data monitoring committee. This is when we had about 208 people in the study. The take-home message from this slide is that we have very broad inclusion criteria. We don't look at a subset of people with MND. There are necessarily some exclusions based on safety of the drugs, but our overall screen fail rate has been 6%, which has been much lower than in historical trials of MND, where a number of people fail the inclusion. The baseline characteristics, we were quite reassured, has been representative of the whole MND population, and that's because of our broad inclusion criteria. We're not restricting inclusion to young people or to people with early disease. So one other concern was that we'd have a whole bunch of people who were living with MND for a long time who might enter the study and skew the results in one direction, but that hasn't been the case. We also have people who are receiving non-invasive ventilation or gastrostomy, and as I mentioned, the drug is given as a liquid, and so it can be given down the feeding tube. In terms of missing data, I mentioned that we're collecting data remotely using video calls, and so we have 99% completeness of our primary outcome measure, the ALS-FRS, and 100% completeness of reporting of deaths. And I mentioned historically as well in trials of MND, people have, it's been a high rate of attrition due to side effects, but really the compliance with IMP has been very high in this study. So attrition in historical trials has been up to 1 in 5 people recruited. And for us, this is from the first 200 people, we've had 12 people who've dropped out, so that's just around 6%. And we have incorporated a number of protocol amendments over the last year which have allowed us to do light touch monitoring of people in the advanced stages or end-of-life phases of disease. So obviously this vulnerable patient group, one thing we want to do is minimise any adverse effects. They have a limited life expectancy as it is. The last thing we want to be doing is causing them more morbidity. And one of the difficulties in this study has been disentangling natural disease progression from what might be expected from the medications they're taking. The bottom line that we've found is that the 2 medicines that we're trialling have not really caused an excessive serious adverse effects in this patient group. So like with many things in healthcare, the pandemic has accelerated a lot of our thinking. We have instituted a number of protocol amendments that have allowed for more remote appointments, e-consenting. We were using bottle returns as a measure of compliance, and we've moved exclusively to patient diaries. One of the things during the pandemic is that a number of research sites had staff redeployed, and so now we have the ability for cross-cover between research sites for remote appointments. We're also moving to use routine blood tests used in NHS care for research screening, and that means that this patient group doesn't have to be burdened with unnecessary extra blood tests when they're having those already. And similarly, if they're having questionnaire data collected, those bits of information can be shared with clinical teams. There's no point putting people through unnecessary cognitive screens if there's going to be no clinical benefit to them. So the Research Ethics Committee liked that idea, and they've actually been supportive through the whole study design as well. We're continuously striving to improve what we're doing, and Emily Bezic, who's here, one of our PhD students who's here today and presenting a few posters on the work that we're doing, we're thinking about how can we gather more data using things like technology, so wearable devices. Could and should we be measuring more than we are? So are there more holistic measures that we should be doing, particularly sort of non-motor symptoms in individuals with MND? And also, what are the factors that influence people taking part in studies? And ultimately, what, if they do drop out, causes them to drop out? And also, how do trials impact on caregivers as well? So there's a lot of work going on in this area, and actually I mentioned that we're trying to move away from the sort of pen and paper based approach of gathering information to more real-time data capture, and we have sub-studies running at the moment considering some of this, and also separate standalone studies in this group of individuals looking at wearable devices. So one of the sub-studies that, shortly after we launched, we realised that people might, there were lots of people who were very, very keen to get into the study and then dropped out early on, and we thought, well, that's a bit strange, what's going on here? What are the reasons that motivate people to take part in the study, and then sometimes what motivates them to leave? So we've set up a sub-study, which is really the first prospective study investigating these reasons in caregivers and people, and I think this one's recruited just under 50 people so far and is ongoing. So, work in progress. We're probably going to cap the number of sites in the UK at 20, just so that we can provide adequate support to them. We're opening in Cardiff, I mentioned, another site in London, and we'd like to open in Northern Ireland as well, and then we'll have good coverage everywhere. We have a statistical amendment to the protocol that actually allows us to reduce the number of participants in the study, and this is a bit of statistical learning from COVID trials, actually. The next thing is the shortlisting of new drugs, and that's in the advanced stages of selection. I mentioned the first interim analysis will occur when 50 participants have been followed up for 6 months or more in each arm. That analysis will be occurring early next year, and there'll be meetings of our data monitoring committee and the trial steering committee. We're always looking to support new sub-studies, and we're doing a lot of biobanking. One thing I haven't discussed is biomarkers, and that's a whole area of a talk for itself, and an area we're thinking about. So ultimately, the way that this platform study is designed is that we can introduce new arms, and we very much plan to do that whether an arm stops or not next year. That's the plan. Good. So I think I'll stop there, and just the summary points are that we're really trying to innovate how we test, how we select new drugs and how we test them. This new platform approach allows for many more medicines to be tested more efficiently, learning from cancer and learning from COVID-19 trials. We're very keen, and we've taken this approach to have an opportunity, provide opportunities for people living across the UK with MND to enter clinical trials, and also work with people with MND to innovate in how we deliver the IMP with remote and community assessments and liquid drugs. Of course, these sort of efforts take up time from lots of talented people, so we have a very active trial management team, trial management group, steering committee, and we've been very well supported by our sponsor at the University of Edinburgh and all of the sites across the UK. Thanks for listening.
Is this one on? Yeah. Well, thanks very much for that, Suvankar. Again, nice presentation and taking us through very clearly the developments that have gone on in the MND SMART trial. Any questions from the floor for Suvankar? No, or any on the chat? No, no. Well, again, another one that's getting off lightly. Obviously, the attraction of coffee is overtaking everyone. But I do have to say that you know, having worked on the M&D Smart study as a non-neurologist, it's been a very interesting learning experience in many ways, both for myself and for the staff who are involved throughout the network across Scotland. Firstly, in the sheer enthusiasm of people to be part of the study. Secondly, their willingness to continue the assessments over time. And thirdly, how well all the staff have adapted to a new set of skills which have been essential to get this study going and maintained. So I would say that there are certainly many lessons that can be learned from that, and it would be nice to see in the other disease areas that we work on a similar high proportion of people coming forward to go into studies. But that's a talk for another day. So I'll once again thank Suvankar, and then the next stage in the programme is to have a short break for coffee, and it is now, well, we're bang on time, which is really excellent. So when do you want to restart, Emma? 20 minutes, fine. So once again, it would be really helpful. I mean, quite happy to talk around in the room, but given that there's quite a constricted space around where the coffee is being served, and given the demands of COVID for ventilation and circulation, it'd be useful if you could bring your tea and stuff, tea and coffee back in here and network in this room rather than outside. Okay, thanks very much.